Okay, so chapter 17, immunodeficiencies. And remember, as a rule, whatever we have discussed so far was normal immune function, and any deficiency in any part of immune system probably will lead to a disorder or a disease. And most of the time it's obvious at birth, and children suffer from it. So most of the time, pediatrics, that's where you'll see immunodisorders. And a lot of work, because there are babies involved, pediatrics involved, and the infant mortality rate used to be very high before vaccination started. So a lot of research has gone into it. And I don't know whether you can recognize that, but this basically is a fruit fly. It's a fruit fly, and many a time some of the experiments, of course, are not done in human beings, and they are done on an animal model. And one of the most important model that we normally see today for especially tall, deficient fruit flies. You remember we talked about pattern recognition receptors, and I said that has to basically be expressed, and that will take care of infections. So if you see a newborn baby having repeated infections, so we need to figure out what's going on, and in this case we have a model that pretty much depicts a tall deficient fruit fly, and that's what it is, because if you are deficient in this particular receptor, you will be prone to some of the microorganisms, like in this case, this fruit fly is prone to a fungus. And interestingly enough, you can see 75% of the known human genes that we've been talking about so far are recognizable match in the genome of fruit fly. So many times when you think of immune system, I'm pretty sure you're thinking of human beings. You're not probably thinking of animals. You're not thinking of insects. And I'm pretty sure that you're not thinking of plants as well. But all of them have an immune system. And all of them have an innate immune system, acquired immune system, and pretty much same lines of problems that we normally see. And you can see this particular fruit fly is being used for doing human research, especially neurodegenerative disorders. So we are working on some of the problems that we have: Parkinsonism, Huntington's chorea, spinocerebellar ataxia, Alzheimer's disease, and many other scientific problems by looking at mechanisms for this particular fruit fly. And most of the time what we have noticed is that these animal models are depicting pretty much a good study tool for us if you want to look for aging and oxidative stress, if you want to look at immunity, diabetes, cancer, and even drug abuse. So you'll see from here, it's difficult to do experiments on human beings, so we have to have a model. And classically, we used to use rat and mice, and then again, with this fruit fly, it's easier to do some experimental plans. So today, I'm going to talk about primary and secondary immunodeficiencies. This primary and secondary word pretty much is the same that you saw in endocrinology. You say primary pituitary problems or secondary pituitary problems. And I'll also talk about neoplasms, cancers of the lymphoid system. Though we are talking of immunodeficiencies, but remember, if some of the immune system components are lacking, so you would not get immunodeficiencies alone. You will end up having neoplasm, meaning cancers as well. And then finally, most of the time we do notice that these children and these babies who have a deficient immune system, they are prone to viral infections. And the typical example for a person prone to viral infection is AIDS, because AIDS virus infects CD4 T cells, and if you don't have CD4 T cell, you basically are prone to infection. So keep in mind that newborns, they don't have well-developed immunity, and we start ASAP their vaccination, but still for some of the vaccine we have to wait for 2 years, and we have to give booster doses. So they are vulnerable to all these infections. And if you see, there is a trend in the newborn, especially repeated infection, and the chances are they are not responding to antibiotics. So we may want to investigate them for primary or secondary immunodeficiencies. Now, also keep in mind that, as I said earlier, that the whole immune system is there to protect you, to defend you, not only against microorganisms, but also against neoplasms, also against autoimmunity, and so on and so forth. So if one of the components is deficient, so that would imply that you are prone to cancers as well. So that's another important thing that you have to keep in mind when we talk of immunodeficiencies. Now, physiologically, we do divide them into primary and secondary. And when we say primary, what we are trying to say is that the deficiency is the cause of disease. So deficiency itself is a disease. And most of the time is hereditary or acquired. And the good thing is that generally it's rare, but again, it's there. So we need to come up with some immunotherapies to compensate for whatever is missing. And when we say secondary, so secondary basically is a result of another disease. I just gave you an example for AIDS. So the person is suffering from a viral infection, and that viral infection will infect CD4. So all the functions of CD4 T cells will be lost. So you will end up having a secondary immunodeficiency. Okay? Now, if you look at some of the tables in your book, and I've just chosen book as a easier way out for you because you can relate and you can also read the book. And pretty much everything that we have discussed so far individually can be compromised, may have a problem. So we can imagine that primary immunodeficiencies can stem from T or B or both. And sometimes there is a problem of phagocytic cells, some of the problem NK cell, complement, you name it. So pretty much all those 10 lectures that we have, each one of them may have a problem. So if you have a problem, and since immune system works in a cascade, so if you compromise one, you're going to compromise the next one as well. To such an extent that even if you have cytokines and their respective cytokine receptors are messed up, or there's some problem with those receptors, again, the function of those receptors would be compromised and you will end up in a problem. Now, the question is that when do we suspect immunodeficiency? And those of you who have had siblings or kids, they know very common for the kids to have recurrent bacterial infections. It's very common, very, very common. So at that time, it's a job of a pediatrician or neonatologist to make sure that he or she is not inadvertently giving antibiotics to those babies. And we have properly investigated for some of the immunodeficiencies that we find. So the most important thing for record purposes, if there is a recurrent infection, that should ring a bell. Especially bacterial otitis media. So they have this middle ear infection, very common. And I don't know if I did teach you ear, but when we talk of infection of ear, it's so common that the middle ear gets infected and then the pus accumulates in the middle ear. And you know the compartment of the middle ear is going all the way, and it's the roof, basically. The roof of the middle ear is the floor of the brain. So there's a chance if pus is there, it can pass and go up in the brain. And many a time the challenge is the antibiotics may not reach there. So what we normally do is we do an operation called laryngotomy. So we poke a hole. So what happens is, if you remember, repair by primary intention, first intention, or secondary intention, when pus accumulates in the middle ear, it's going to rupture the eardrum. And when it ruptures the eardrum, it's going to create a very nasty or an irregular kind of a lesion that will be difficult to repair. And if you remember, when we give a surgical cut, the margins are approximated, so that is considered as a clean cut. So the idea is that instead of babies going through otitis media and getting their ear ruptured and have a nasty scar and then replaced by fibrosis, so let's poke a hole and put a small tube there. So they put like a small tube over there. So whatever is there in the middle ear gets drained out. And once they grow up, for like a couple of years, they will remove them out. So I'm trying to kind of tell you that this is very common, but keep in mind that you have to investigate for immunodeficiency. So bacterial pneumonias are important, and this basically is coming from B or antibody deficiencies, so that's important. The other important thing is that if anyone is susceptible to fungal, protozoal, or viral infection, normally if you are adult, your immune system should be good enough to take care of most of the fungi, protozoa, or viruses. But if you do have a T cell or cell-mediated immunodeficiencies, you're going to fall a prey to these diseases. And then again, if a person gets systemic infections, remember, as I said earlier, as a rule that antibiotics is not an option for the amount of bacteria that we fight on a daily basis. And this is going to be my first lecture when I talk about bacteriology. Why is it so? So if a person has a systemic infection and that is coming from a bacteria which normally is of low virulence, suggesting that this bacteria should not cause a problem to normal person with a normal immune response. If such a thing happens, then we should be looking for immunodeficiencies. And then again, recurrent infection, opportunistic infections, this again would suggest that your immune system is lacking in some of the component, whether it be a cell-mediated wing or a humoral wing antibody. So any of these can be compromised. If you look statistically, again, 50% of the problems that we see in primary immunodeficiency is antibody deficiencies. Then you can see 20% are combined antibody and cell-mediated immunity, 80% have phagocytic disorders, suggesting to you that, remember, for even antigen presentation, you have to have antigen-presenting cell phagocytose those particular microbes. So if they have a problem of phagocytosis, they would not be able to present the antigen to T cell. And then again, your immunity will be compromised. Then we have 10% disorder for cell-mediated immunity alone, 2% complement deficiencies, and then again, also keep in mind that there may be some genetic defects. When we make those B cells and T cells and we go through the sequence of cell cycle, there may be a problem, okay? So this is pretty much a rough percentage of what we normally see. But again, the most important thing is that 50% of all primary immunodeficiencies are coming from antibody deficiencies. And again, also keep in mind that other than IgA deficiency, the frequency is generally low because it is there in the kids. Now, this was a slide that we did discuss when I was talking about where the cells are produced. So you can see from here, right from the beginning, when the cells are being produced, so B and T cells are being produced in bone marrow, right? And then T cells are educated in thymus. So each and every step that these cells go through, you may have an associated problem. For example, in this figure, if you pay attention, that some of the lesions are presented as circle, which suggests a combined immunodeficiency, both B and T. And some of them are suggested as triangle, suggesting that they are T cell disorder, and some of them are in square, suggesting to you that they are B cell, primary B cell triggered. And you can see from here, this is a key. And you can see these are some of the disorders that we clinically see. You don't have to kind of remember them all, but what I want you to do is that keep in mind that when a lymphoid cell starts going through a phase, so at any step of proliferation, growth can be arrested and can be blocked. And that basically could lead to a disease. For example, if lymphoid stem cell are not making pre-B cell, so what will happen is that you will have a severe combined immunodeficiency syndrome. You can see from here, right, number 1. Okay? And again, by the same token, if the lymphoid stem cells are not making pre-T cells, you may have a congenital thymic aplasia, DiGeorge syndrome that we suggested earlier as well. So the whole idea is that when we move from one phase of a B and T cell to another phase, growth can get arrested. And you will wonder, how do we know that? So what we will do is clinically, we'll take a bone marrow biopsy and take all the cells and run them to FACS analysis, because we know for sure the markers for CD4, markers for CD8. We can also go for the markers of some of the antibodies which are presented. Okay? So let's take, for example, a B cell. So B cell is there, it's a mature B cell. And then again, you can see even after a mature B cell starts making different types of antibody, each and every step can be compromised, because it's not showing over here because there's an enzymatic activity involved, a lot of steps involved over there. So we have removed them for brevity. But the thing that you need to understand is that when the cells go through proliferative phases, each and every phase can be arrested. Now also keep in mind, when we talk of cancers, for example, you must have heard blood cancer. So when they say blood cancer, it could be a B cell cancer, it could be a T cell cancer. If you arrest, for example, or block proliferation of a B cell over here or T cell over here, so what will happen is that the cell which is released into bone marrow or circulation is not functional. So it would not do what it's supposed to do. Plasma cell will not make antibodies. T cells, if they don't have a proper MSC, they would not be able to talk to other cells. Okay? So you can see from here that, for example, let's pick up a selective IgA deficiency, which is like at number 6. So if the mature B cell is unable to make IgA, so what it would mean is that your mucosa of whole body will be vulnerable to infections, because IgA is required in your mucus membrane for protective action. That will be a big loss. Now, if you pay attention to number 7 over here, when you have a lymphocyte, so let's assume this lymphocyte should have picked up a CD4 marker or CD8 marker or all other receptors, and they are not showing this receptor, then we have a disease we call bare lymphocyte syndrome, suggesting to you that they are kind of naked. There is no receptor expressed on the surface, so they pretty much would not engage and then do the normal function. And then again, number 9 suggests to you defects in T cell signaling. And then again, T cell signaling, remember we talked about outside in signaling, all those enzymatic steps, they're all proteins. Proteins are regulated by genes. There could be genetic disorders that can basically arrest and cause immunodeficiencies. Okay? Now, severe combined immunodeficiency we call SCID. And in this case, you will see that most of the time the presentation is that lymphocytes don't have those receptors on the top. And if you remember recombination activation genes, which are required for picking up appropriate heavy and light chains, they have a problem. If, for example, they have other Omen syndrome, which I'm going to discuss in a while, and Wiskott-Aldrich syndromes, these are two of the syndromes that we normally see clinically. And the problem with this one is that graft versus host disease. So if you want to graft or give a organ to a donor, the recipient will reject it. So it's a typical disease we call graft versus host disease. And then again, it's coming from immunodeficiency that we normally do. Now, other than some of them which are mentioned over here, I don't want you to go in detail, remember all these things. But what I want to emphasize on you is that all the problems that we see in terms of deficiency, there are tons of them, and they basically are coming from the genes. So you can see all those genes that we were discussing about, variable, hyperdiversity, all these genes are deficient, and there is a problem there. And especially Omen syndrome, which is RAG1, RAG2, and then Bruton's kinase, X-linked agammaglobulinemia is very common. And then again, you will see over here some of the protein signaling pathway that you saw in one of the figures, like ZAP70 or protein kinases and so on and so forth. They can all be deficient. So what will happen is that you will have a immune defect. So you can see all those immune defects over here. They could be right from absence of B and T cell, absence of antigen presentation, absence of making antibodies, and sometimes what happens is that you end up in a clinical symptom because your spleen gets enlarged. Your liver gets enlarged. So that's again hepatosplenomegaly that you normally see over here. And then again, with all those defects that you normally see over here, the price that you pay is susceptibility. And I'm going to take you through some of the questions that you may expect over here. For example, when in roadside accidents, when spleen is ruptured, we remove the spleen because spleen bleeds a lot. And if these patients' spleen is removed, the condition is asplenia. So what this means is that now they are susceptible to encapsulated extracellular bacteria. That's what it is. So we have to make sure that we give them vaccine for that. Okay? Chronic granulomatous disease is called CGD. It's important because if you remember your electron chain, and if you remembered all those cytochromes that you study in biochemistry where you have NADPH oxidase where energy is produced, so that pretty much, if it's impaired, it's going to impair neutrophil function, and you know neutrophils are the first line of defense. If your neutrophils are non-functional, so you will have problem of bacterial and fungal infection because you lack that particular enzyme that is required, NADPH oxidase, and the condition is called chronic granulomatous disease, CGD. So that's something important. Okay, I talked about SCID because in SCID, the regulatory enzymes which are RAG1 and RAG2 are deficient, and then you are pretty much prone to all infection. And Again, you can see from here, ZAP70 deficiency, that was also important. And the rest pretty much are easy. All you need to know is if there's an IgN deficiency, IgA deficiency, you know what is the function of IgA. So you pretty much can start thinking as to what will happen to a particular patient if his or her immune system is compromised. Okay? So you can see from here, the whole picture is infection, 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 infection. As I said over and over again, that the amount of challenge, the amount of bacteria that comes and hits our immune system is too high. And we need to have a very patent immune system. If your immune system fails, then again, you will have all these different problems in terms of bacteria, viral infection. On top of that, as I said, that you'd say, okay, what's the big deal about that? We can give antibiotics, okay? How long are you going to give antibiotics? You cannot give antibiotics for the whole life. If you give tetracyclines, for example, so tetracycline are going to knock your bone marrow. So you have another problem. Some of the problems are coming from the overuse of antibiotics, where you use antibiotics and they will shut down your bone marrow. Sometimes you lose your platelets, agranulocytosis. So you can see from here, infection. The other two important things which I hope that you noticed when I said that when your basic players are deficient and they have a problem, not only you will be prone to infection, but your ability to fight cancer is also lost. And they themselves will cause a cancer, because if the cells are not controlled and you basically lose that regulatory protein, so you may have autoimmune diseases. So you can see over here, autoimmune diseases. So once we investigate, and it's a very common question asked, that if you do find a baby who has infectious diseases, immunodeficiencies, what other tests you want to do on that? So we are also looking for autoimmune diseases as well. So you can see autoimmune diseases over there. And sometimes cancer. So immunodeficiency, autoimmune diseases, and cancer can be paired up, because there is a problem, and you wonder why. Because we have to have a patent cell-mediated immunity as well as humoral immunity to fight and fix the problem that we normally see. Okay. Another example, genetic linked diseases. Again, as I said, primary immunodeficiencies are number one genetic. So if it's running in the family, it's going to be there. Now you can see from here, this gives you an example, as I said earlier, a cytokine receptor. So cytokine receptor, if it fails to generate, if the signal is not there, remember when I said you have to have a ligand for all the major Th1 and Th2 cytokines. But these cytokines basically, like IL-2, IL-4, 7, 9, and 15, they would need to bind to a particular receptor. Like in this case, there is a gamma chain over here. So gamma chain has to be there before the cytokine will act and then perform whatever it needs to perform. But if you have deficiency, like if you have a missing gamma chain, so what will happen is that there will be no intracellular activity. So the purpose of that particular cytokine is to do something, and that will be lost. Okay? So cytokine mediator receptor signaling, that's what it is lacking. So you will not be able to transcribe those specific genes which are required. And this basically is X-linked SCID disease that you normally see over here. Okay. The other example is that if, as I said earlier, you may miss a membrane determinant. So there is a T cell or there is a APC, and you pretty much know by now that they have to express what they need to express, and they would at least need to express is at least one receptor to get something going. So you have, for example, a macrophage, and this macrophage, in order for presentation of antigen presenting a antigen, it needs to present it in context with MSC class 2, and that is deficient. If that is deficient, so of course T cell will come and is not going to talk to it, and there will be no activity. Or what may happen is, for example, T cell itself has to express those co-stimulatory molecules or adhesion molecules and all different molecules, and they are proteins. And these proteins basically are under genetic control. So they are coming from your genes. Now, if your genes have a problem, so they would not express, for example, CD40 ligand will not be expressed. And you know the importance of CD40 and CD40 ligand is that this is a connection you require for the antibody switch. So there will no be antibody switch. So they would not produce the required antibodies. And then again, they may also sometime present, because they're proteins, mutated signals. If those signals are mutated, and then again you will see the cells would never recognize. So the normal process of signaling will be lost. Right? And as I said earlier, since most of the receptors are proteins, and proteins are under tight control, they have a genetic component, and then we can trace back and find out the cause. But the problem is that many a time there's nothing you can do. There's nothing you can do. What we are trying to do these days is that sometimes if you have a deficiency of a particular gene, so what we normally do is that we have a technique, we call it gene gun. So we put that gene that is deficient and put it in a system and kind of shoot it into your cell, so that your cell will pick up that particular gene and incorporate into the system. But again, these are experimental studies, and many times it's difficult to even experiment on human beings, because if you want to go for one gene, you may end up causing problem to the other gene. But we know for sure that they are there. Okay? And what do you think if I was to tell you that CD40 ligand is mutated? So if it's mutated, it's not normal. It would not talk to CD40, and then you will end up having a hyper IgM syndrome. Why do you have a hyper IgM syndrome when there is a mutation of CD40L expressed on a T cell and it needs to talk to CD40? Anyone? Correct. Simple. Because class switching will never take place. If there is no class switching taking place, so you are making IgM. You're not making any other than IgM and IgD. So you will have a problem we call hyper IgM syndrome. Good. Simple. So that's what you need to understand, the basic concept, and it's easy for you to go back and link it with the problem is. Okay. Now, this slide tells you a typical TCR, alpha beta, and then again we have a zeta chain attached to it, and if you remember the CDR complex. And then when the activation takes place, there was a molecule I said earlier, ZAP70. So ZAP70 gets phosphorylated. So in this case, for example, if you have a gene deficiency of making a ZAP70 which is deficient, so you will not have a proper antigen processing by your TCR. So TCR will just be there, capture an antigen, but remember that is the first signal, and you have signal transduction molecule in terms of zeta chain. So you have to send a signal in the cell, and this cell will be translated into proteins. Everything has to go to the nucleus before the synthesis of that particular cytokine take place. So you can see that's another example. If you have deficiency of those molecules over here, and these are ITAMs, so ZAP70 would not bind to ITAMs. And again, this is a good information because I'll give you some of the drugs today at workshop that are new drugs for asthma, allergy, cancer. They're all working on intracellular pathways. So it's for your understanding as well, you pretty much get a good hold of intracellular signaling pathway. Okay. Now, this is an example, a simple example we already said. There could be problem with some of the ligands. For example, you need neutrophil to come and fight infection in the tissue, and neutrophil is running in the blood. But remember, when we talked about neutrophil, what causes neutrophil to leave the blood and go to the tissue, right? So what happens is that there is a receptor expressed on neutrophils, and there is a corresponding receptor expressed on the endothelium. And you can see E-selectin, and mostly over here, these are integrins. So if this is missing, right, or if one of the chain is missing, you would not have a proper attachment of that neutrophil to the endothelium. If that happens, neutrophil will never ever come. So you'll have a boil over here. Normally you have a boil, and then you see a pus there. So what has happened is that neutrophils have passed from your blood, passed through your endothelium, have reached those particular site and killed bacteria. So that pus is dead neutrophils and dead bacteria. So imagine if you don't have that, so what will happen? What kind of problem do you expect if the neutrophils don't go and kill the bacteria in your tissue? What do you suspect to happen? Infection is already there. Sorry? Septicemia, very good. She's right. Because you have a local infection, so neutrophils would have done a local job, killed it there. If neutrophils were not there, this infection will spread in the whole blood, and as she's saying, septicemia. So we have a bigger problem there. Yeah. Correct. Does that make sense? So you can see from here, so there are different diseases. For example, this one over here, we have, if there is a missing selectin ligand altogether, or if there is a missing chain, right? And this again will lead to neutrophils not getting into the system. Okay, let's see, everything is okay, there's no problem, neutrophils are okay, so there is no adhesion, leukocyte adhesion deficiency. So leukocyte adhesion deficiencies, LADs, is not there, and neutrophils go over there. And they are able to even phagocytose. So what is the next step in a neutrophil when it phagocytoses bacteria? What do you expect now to happen? Sorry? Very good. Now lysosomes have to go and attach to that phagosome. We call phagosome-lysosome fusion. Exactly correct, good. So lysosome has to come over here, you can see, and take care of phagocytosis. And many a time it releases what is a typical killing mechanism the neutrophils have. How do they kill? Say it again? Say it again? Peroxide. Peroxidases, basically. Correct. These are reactive oxygen intermediates. Remember, many times you want to take antioxidants? So you take antioxidant because you don't want your reactive oxygen intermediate to kill your cells to make you look old. That's what it is coming from. But in this case, neutrophils have many different things. One of the things is they have oxidant. And remember that where is that oxygen coming from? Where is that oxygen that you are using to kill bacteria is coming from? Which place? mitochondria, and electron transport chain. That's where you generate oxygen, correct? Again, your Krebs cycle. That's basically a clinical implication of your Krebs cycle. So what is happening is, in this case, you have a problem of NADP oxidase. That is an enzyme required to produce that oxygen radical. Okay? So what will happen is that your neutrophils will still phagocytose but not kill. And we call that as a chronic granulomatous disease. If, for example, they are unable to phagocytose, that's another disease, Shadiak-Higazi syndrome. But you can see each and every, there could be multiple problems there. And we have to understand the problem before we treat them. So every patient is different. And especially when it comes to babies, you have to make sure that pediatrician sees it. So you cannot give over-the-counter advice. And many times in many cultures, people will just go take antibiotics for everything. That's not correct. And it's not going to solve the problem. So you have to understand the whole immune system, figure out where the defect is, and then go for the repair. And if you don't understand that, then it will be difficult for you. Okay? So far, so good? All right. So the whole idea for this lecture is I just want you to think of the basic immune response and then figure out on your own how and why things go wrong and what are the possible fixes. I'm not talking about the fixes right now, but you can imagine what will be the possible fixes that the options that we have. Now, we talked about leukocyte adhesion deficiency, LAD, we saw that. And you can see that if that is there, you will have a widespread infection with capsulated bacteria. Right? If you have a chronic granulomatous disease, I told you that you cannot produce O2, I just told you. And because the enzyme required for electron transport chain, which is NADPH oxidase, is the cytochrome. That is deficient. So you will have impaired killing of phagocytosed bacteria. Right? So babies have chronic bacterial and fungal infections. And then other kind of, like for example, myeloperoxidase deficiency. So you can see from here, as one of your colleagues just told me, that the moment bacteria is taken in, that particular chamber is called phagosome. And the next step that would occur is that lysosome will come and fuse with phagosome. Now you can see from here, if that fusion is deficient, again nothing will happen. And you will guess, why would that happen? Can somebody guess that what would cause a lysosome not to come and attach itself with the phagosome, other than genetic disease? Just guess. Say it again? Correct, but what? Okay, let me rephrase it. So, some type of drugs, not really. This is in the interest of some of the microorganisms, like Mycobacterium tuberculosis. They have a clever plan, so they probably know immunology than we know, probably. So they know that that's what is coming up. So what they would do is, they would not allow phagosome-lysosome fusion. So they will go and sit in your cell forever, never to be found out by your antibodies. And they, remember I said different routine. What was the difference I told you between neutrophil and monocyte or macrophage? The other day I said that, remember, a major difference. Sorry? Antibody? Okay. Say it again. Size? Well, I said that wasn't a big issue. Say it again. Exactly. Very good. Long live and short live. Because neutrophils have a half-life of 72 hours. So if a bacteria enters the neutrophil, either way, neutrophil is going to die in 72 hours. Not a big deal. But if a bacteria goes in a monocyte, and that bacteria We call that mechanism intracellular survival. I'll discuss that in detail. But just to give you a heads up on that, that the reason we are unable to control Mycobacterium tuberculosis is because he's smarter than us. It would not cause phagosome-lysosome fusion. It's going to go in your macrophages, and macrophages can lodge anywhere in the body till you die. Or it's going to come back on you when your immune system goes down, it finds this opportunity, and then going to cause disease. Make sense? Okay, now you start thinking. Very good. I didn't realize that you just have to stress you with the first test and everybody starts thinking. It's a big change, I just noticed that. Good, well done. Especially the first 3 rows. They've been answering my question. Anyway, complement cascade. Again, remember, you would think who on earth would want to know about complement? But remember that you're going to deal with complement because you need to have all these alternative pathway, classical pathway, and complement. And if there is any deficiency, for example, if there is a deficiency of the complement syndrome, so you will have infection with encapsulated microorganisms. Why would you have a problem with encapsulated bacteria versus a non-capsulated bacteria? Why would you have a problem if you have a problem with complement? Why we are talking about a bacteria with a capsule? Raise your hand and ask, yeah. Well, you just tired stretching? Okay. Because remember that any bacteria that has a capsule has to be opsonized. You have to sugar-coat it. You have to put complement. If there is no complement, you have receptors CR1, CR2 in your phagocyte, they would not engulf it. So you enhance the uptake of opsonized bacteria. You sugar-coat it. You increase that thing. So you can see from here, that's the reason. Okay? And then again, you will see one most important thing that I will discuss when I move on to sexual transmissible diseases, and we have a workshop on that as well. Sometimes if you even have a deficiency of those particular convertase enzymes. So typical example is that you have a problem from the complement cascade from anywhere from C5 to C9, right? And you are not involved in any promiscuity, or you're not into that sexual transmissible high risk criteria, and all of a sudden we see that you have Neisseria, and that is a STD. Now, with all due regards, we don't want to question your honesty in telling us the proper history, but what has happened is that you have a complement deficiency. So that pretty much saves your reputation because you have a complement deficiency. So there you go. Okay, so complement again, let's see on this slide. C1, C2, 3, you have immune complex disease. If you have a C3 problem, because C3 is the major. When I taught you complement, what did I say when I said C3? What is C3? Yeah, major opsonin. Major opsonin. We do C3 levels. Is a major opsonin. And then C5, C9, we have Neisseria infection. And yet you can see that many other problems that we normally get when you have complement problems. And we want to investigate that. Few minutes on secondary immune deficiencies. I'm going to take secondary immune deficiency separately as an AIDS lecture full-fledged. But people may have temporarily transient immunodeficiency if you put yourself into malnutrition. So if you don't take calories that are required, and if you all of a sudden want to become vegetarian and no meat at all, that you don't compensate for the lost proteins, so you basically are messing up your immune system and you may have immune deficiencies. And then again, drugs, anti-cancer therapy, immunosuppressive drugs, and many other things are there. And all autoimmune disease patient, all cancer patient, because their whole immune system is fighting cancer. Whole immune system is fighting bacterial infection. So you have a generalized kind of immunodeficiency. So that's another important thing that is there. But the common cause that we normally see, for example, you are in a, I don't know, on a diet program or a weight reducing program, and you're not wise enough to pick up what is required in terms of your proteins, because you have to have these proteins produced. So you put yourself in a malnourished picture, right? And your immune system basically goes through immunodeficiencies. You will not be making the IgA antibodies and many other antibodies that are required, because they are all proteins. So remember, whatever you do, at least make sure that your diet does have appropriate portion of protein, because your immune system is going to depend upon that. And then again, as I said earlier,